My name is Rebecca Landerkin, and I will be presenting the vitamin C concentration and the effects of pasteurization lab. So first we'll talk about vitamins. They are small organic compounds that are essential for cell growth, function, and development. They're going to facilitate some of the chemical reactions that occur in the cell that makes them catalytic in nature. So they're needed in small quantities, but they cannot be synthesized in nature by the organisms that need them. So they can be consumed through diet or, if need be, supplements. So there are two categories of solubility for these vitamins. They're going to either be fat soluble, so they're stored in the fat cells of the organism, or they're going to be water soluble. And those vitamins, if they're in excess, they're going to be flushed immediately by the organism. And vitamin C is going to be one of those water-soluble vitamins. So vitamin C is essential for the cell to be able to absorb iron. It's essential for skin, bone, and connective tissue health. So if you have a vitamin C deficiency, you might see some bruising, you might have bleeding gums, all signs of scurvy, which is vitamin C deficiency. So we need as much as we can get, and especially in the raw form. That is because heating or cooking of vitamin C will destroy that particular acid, the ascorbic acid. So, because cooking destroys the vitamin C, in turn, so would pasteurization, because that's a process that is going to heat up a liquid for an amount of time in order to eliminate all of the bacteria in that liquid. So that process would definitely decrease some of the vitamin C levels in, let's say, orange juice that's getting pasteurized. So my group, we hypothesized that the vitamin C levels will be significantly higher, in the fresh orange juice compared to the pasteurized orange juice. So how we did that, we took a single orange, we cut it in half, we juiced both halves, and we allowed that juice to mix. And we strained that juice through 4 sheets of cheesecloth. That way you were eliminating all of the large particles from the juice. We decided to go with 3 replicates for the pasteurized samples and 3 replicates for the fresh. That way we were able to measure some of the variability within those replicates. So we placed about 1.5 milliliters of juice into the 6 of the microcentrifuge tubes. And at that point, we centrifuged the 6 samples at 12,000 RPMs for 2 minutes so that we could separate the remaining solute within the juice to create a little pellet at the bottom. And once we transferred that supernatant into new centrifuge tubes, we then set aside the 3 to pasteurize and the 3 to sit and wait. So the 3 that waited, they just waited on an ice bath. The ones that we pasteurized, those went into a water bath that was 93 degrees Celsius for 2 minutes, and then another bath for 80 degrees Celsius for 20 minutes. And after that was done, we took those pasteurized samples and we placed them in an ice bath for 5 minutes. So at this point, we're getting to an organic dye known as DCIP. And so that is something that's going to be able to measure the acidity, the acidic value of a liquid. So if an acidic solution is added to this dye, it's going to become a colorless solution when the dye is usually blue in color. So the more acid you have in something, the clearer the liquid is going to get. And we're dealing with ascorbic acid, which is vitamin C. So the more vitamin C we have in a sample, the clearer the liquid is going to be when in contact with the DCIP. So we created we didn't. Our professor provided us with a dilution rate of 1 to 5 for testing the absorbance of our samples. So the juice was added to distilled water, and then we added the dye as well. So at that point, the mixture needs to be mixed, and we put paraffin wax on the centrifuge tubes to mix them. And we then read each of the absorbance rates through a spectrophotometer. And over here is where we have those absorbance rates or values listed. And here you can see it supports our hypothesis. The fresh juice was significantly higher in vitamin C compared to the pasteurized juice. So that was one thing that we took note at first. And so even up here, we have the assay graph for the vitamin C concentration, which was also given to us by our professor. And there are some other data points not shown in this graph, but there's really no way to measure those. Once you get a certain absorbance, there's just too much vitamin C, so we zero in on a few data points so that we can clearly measure our vitamin C concentration depending on the absorbance. And the slope is negative, so it's kind of backwards. The lower your absorbance is, the more vitamin C you're going to have in your sample. So again, we have the higher absorbance rates lower, and then the lower absorbance rates having higher vitamin C. So I don't know if I said that right, but the absorbance is going to be lower for higher. And compared to the other groups, it was a little interesting. So for our absorbance rates, the difference between fresh and pasteurized was about 12%. There was another group, the Love group also had about an 11%, but the Padilla group only had a 1% difference between their pasteurized and their unpasteurized. And what that means is that the pasteurization process had no effect on the vitamin C levels in their case. And they also had 5 replicates for their experiment. So that made us consider maybe we should have had more replicates. We should have been testing more to see a better variability between our data points, because our data points only have about a 4.9 difference in the pasteurized and a 4.7 in the fresh. So in conclusion, we decided perhaps more replicates in the future, and better care with pipetting, because perhaps maybe some of the pellet was transferred. Perhaps the cheesecloth didn't get all of the larger molecules that could have been collected, or those particles, or maybe some of the pulp did get transferred. So there could have been a few number of issues. But all in all, our end result supported our hypothesis.